Hello everyone! In today's video, we will talk about DNA replication.
DNA is a molecule that is made up of two strands twisted around each other in a double helix shape. Each strand is made up of four chemical bases represented with the letters A, T, C, and G. The two strands are complementary. This means that wherever there's a thymine, there will be an adenine on the opposite strand, and whenever there's a cytosine, there will be a guanine on the other strand. Each strand has 5' end and a 3' end. The two strands run in an opposite direction. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate each strand. This unzipping is done by an enzyme called helicase and results for a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks a starting point for the construction of a new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The other strand, or called the lagging strand, cannot be made in this continuous way because it runs in an opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragment. Each fragment is started with an RNA primer. The DNA polymerase then adds a short row of DNA bases in a 5' to 3' direction. The next primer is added further down the lagging strand. Another Okazaki fragment then is made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another polymerase enzyme then fills in the gap that are left behind with DNA bases. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand and one new one.